Okay, hello everyone. My name is Kelsey Rosales-Torres. I am majoring in neuroscience. My faculty mentor is Dr. Wendy Saltzman of the Evolution, Ecology, and Organismal Biology Department. And my talk is on the effects of parenthood on neural responses to pup-related cues. To start off with some background, a key term is biparental care, which refers to care by both mothers and fathers. Only about 5 to 10% of mammals exhibit biparental care. Some example species that do are marmosets, shown on the left, prairie voles, shown in the middle, and humans, shown on the right. Another example species, which is the species that our lab studies, is the California mouse, Peromyscus californicus. This rodent is naturally monogamous and biparental, and both mothers and fathers exhibit comparable levels of parental care, which includes licking, grooming, and huddling of pups. Mechanisms underlying the onset of maternal care are fairly well understood. However, this is not the case for mechanisms underlying the onset of paternal care. One example mechanism that might have a role is that of neuroplasticity, which refers to the reorganization of neural pathways. And an example of neuroplasticity is sensory plasticity, which refers to neural changes involved in detecting and processing sensory stimuli. And so my study focuses on two main brain regions. And I will use this picture on the right of a sagittal view of a rodent head, the nose being on this side and the brain being on this side. And so the first region that I focus on is our main olfactory bulbs, shown here in green, which is essential for chemosensory detection and has downstream effects on other regions in the brain. One example region being the medial preoptic area, or MPOA, shown here. The medial preoptic area is essential for parental care in mothers and fathers. And this region also receives input from the auditory pathway. And so we focus on these two regions in response to chemosensory and auditory pup stimuli. And so the question of my study is, does sensory plasticity in the main olfactory bulbs and medial preoptic area occur during the onset of parenthood in male and female California mice? And we hypothesized that activation of both of these regions in both male and female California mice in response to pup-related sensory stimuli is higher than in response to control stimuli, is higher in parents than in virgins, and is higher in response to combined presentation of both pup vocalizations and pup sense than in response to either of those stimulus alone. As for methods, we had 29 virgin pairs, which included a male and an overectomized female. Overectomized referring to a surgical process removing the ovaries. And this ensures that the female would not engage in sexual behavior, nor would this pair reproduce pups. And we also had breeding pairs, which included male, a sham overectomized female, and their first litter of pups. And sham overectomy refers to the same surgical procedure. However, these females did not actually have their ovaries removed. And this is done as a control for any potential effects of the surgery. And so each male and female underwent a single stimulus exposure. And these are the four stimulus treatment groups. The first being pup scent, where the mouse was exposed to a white noise, which is the control for the auditory stimulus, and pup cotton, pup scented cotton. And the second group being a pup call group, which was exposed to pre-recorded pup calls, and fresh cotton being the control stimulus, control for the chemosensory stimulus. Third, we had a group exposed to both pup calls and pup cotton, both the pup call and pup scent. And then last but not least, we had a control group, which was exposed to white noise and fresh cotton. And each stimulus exposure test was 10 minutes in duration. And the setup was as shown here. The mouse was placed in a cage with a wire mesh T-ball containing either clean cotton as a control or the pup scented cotton. And underneath the cage was a speaker playing either white noise as a control or the pre-recorded pup calls. And in these pictures, we can see the stimuli being collected. On the left is the chemosensory stimuli being collected by rubbing a pup with a cotton ball. And that was placed in the wire mesh T-ball in the test. And then on the right picture is a pup vocalizing or producing its pup call. And the pup calls were pre-recorded and then played via speaker during the test. And so here's a short video clip of a mouse shown here, exposed to a pup scent on the cotton ball in the wire mesh T-ball and white noise playing in the background. And so that mouse was clearly investigating the scent in that wire mesh T-ball. And so 1 hour after stimulus exposure test, the mouse underwent transcardial perfusion, which is done for tissue fixation. And then we went ahead and took 40 micrometer coronal sections of the brain using the cryostat shown in the picture on the left here. And we obtained brain slices as shown in the right picture, these two slices being main olfactory bulb tissue, and these slices being tissue slices that include the medial preoptic area. Then we performed immunohistochemistry staining for FOS. And FOS is the protein product of the immediate early gene CFOS. And quantification of FOS can be used as an indicator of neural activity and gene expression. Then we imaged the tissue slices that were FOS stained, and we used 200 by 200 micrometer square areas for the quantification. The imaging was done using confocal microscope, and we obtained images as shown on the screen here. The left image is a main olfactory bulb tissue in a California mouse father. And this red box highlights the 200 by 200 micrometer square area that we looked at. And this is a close-up. This slice does not show FOS staining, but if we look over on the right, this is an MPOA tissue. This red box highlighting the MPOA area that we quantified the FOS in. And this close-up shows a few FOS positive stained cells shown by the bright green dots there. And so then with these images, we quantified the FOS using a program to do so. And then last but not least, we did statistical analysis using linear mixed effect model to analyze the FOS. As for results, I will first cover males and then I will cover female results. And I will start with the MOB and then the medial preoptic area. And in all of my graphs, the y-axis shows the number of FOS positive neurons. I show the mean and standard error. And the x-axis shows reproductive condition in the left graph and stimulus treatment on the right graph. And so for males, MOB FOS was higher in virgins than in fathers. And FOS did not differ among the stimulus conditions in males. And in the MPOA, FOS was higher in fathers than in virgins. And FOS did differ across stimulus treatments. And so looking at the graph here, males exposed to pup scent had higher FOS than males exposed to the control, while males exposed to pup call and pup scent had higher FOS than males exposed to the control. And last but not least, males exposed to both pup call and pup scent also had higher FOS than males exposed to pup call only. In females, in the main olfactory bulbs, FOS did not differ between reproductive conditions, and FOS did not differ among stimulus conditions. And in the MPOA, we got similar results. The FOS did not differ between reproductive conditions, nor did it differ among stimulus conditions. So in summary, in females, FOS in the MOB and MPOA was not affected by motherhood or stimulus treatment. And fathers had lower FOS in the main olfactory bulbs, but higher FOS in the medial preoptic area than virgins. And in males, MPOA FOS was increased by exposure to pup scent, especially when the mouse was exposed to both the pup scent and pup calls simultaneously. So some conclusions that we make are that fatherhood may dampen activity of the olfactory system, especially in response to pup stimuli. Fatherhood may upregulate activity of the medial preoptic area, especially in response to pup stimuli. And overall, this is one of the first demonstrations that the onset of fatherhood in a biparental species can be associated with sensory plasticity. I would like to thank everyone listed on this slide and the sources of funding. And I'd like to give a special shout out to Dr. Carrie Wilson for all of her guidance throughout this process and supervision throughout this process as well. And thank you for your attention.